Hello, hello, and welcome everybody to today's webinar on Alzheimer's research and why so many trials fail to deliver actionable insights in this area. My name is Valerie, I'm Wemedoo COO, and today I will guide you through this session. And I'm today joined by two very exciting guests. We have today Professor Dr. Matthias Günther, who is the founder and chief executive officer at mediri, as well as Dr. Nikola Cihoric, who is the founder and chief product officer. So as such, my colleague here at Wemedoo. And today we really have an exciting topic to discuss. But maybe before we start, I also want to invite the audience today, ask your questions throughout the session in the comment field from whichever platform you're watching from today. And we will make sure to address them towards the end of the session today, or also in follow-ups should we not get through all of the questions today. But yeah, with that, let's look at the current Alzheimer's landscape. And, I have a business background. So from a commercial perspective, I've been following lecanemab's, let's say, rather rocky market entry as of 2023. I think right now we're seeing a wave of observational studies. So for sure, there's scientific progress in this area, but that somehow does not yet seem to fully translate into actionable results. And I think that's where we got to this topic of today. So maybe let's start with that, Matthias, Nikola. Why is that from your perspective? What really stands out the most?
Yeah, so from my perspective, I think it's several aspects. One, of course, is Alzheimer is a really complex disease, and it's not yet fully understood, and it involves so many different pathological processes, which are partly known. And if you try to target just a single mechanism, it just often proves insufficient to actually go this way. And because the disease evolves over such a long period of time, it's also very difficult to actually recruit patients in an early phase. Most of the patients we are seeing in the clinics and also in trials are in a very late state already, because clinical symptoms appear in a very late phase. They might have a history of several decades already, and that makes it really difficult to assess, and is a huge challenge for the for clinical trials for developing drugs, in my opinion. And what we will also learn, there are ways to actually diagnose it earlier, or at least have some hints. And I'm really looking forward to share some of those insights. But maybe Nicola, you want to add?
As you rightly said, Alzheimer's disease is really a complex disease of central nervous system, and we have a lot of challenges understanding biology. But beside biological complexity, what we see in clinical trials in Alzheimer's disease is severe data fragmentation. So what that means? You have imaging data in one system, clinical assessment in another system. Then biomarkers result in third system. Then patient-reported outcomes in fifth system, and so on and so on. So when you need to make some treatment decision or assess efficacy, you basically are forced first to put all the pieces of puzzle together that are scattered across 5 different boxes. Additionally, when you don't have integrated biomarkers at the point of screening, when imaging data is collected, they must be rapidly assessed and compared against eligibility criteria in real time. That is something that doesn't really happen. So you have to set up the system and integrate data, and you are wasting enormous resources on patients who are also never appropriate candidates. So all of that add to complexity of biology and reduce efficiency and effectiveness of getting new insights and conducting clinical trial in Alzheimer's disease. So this is additional burden to tackle this disease and move forward.
And even to stay in your picture, I think even within the boxes, we sometimes have quite a mixture of different information. So it's not only to bring the different boxes together, but
Yeah,
it's
exactly.
also inside the boxes to
Exactly.
get some organization.
Great. Thank you so much both for this great intro. And I think you're already giving us lots of topics to dive deeper into and to learn more about. Let's maybe first start really on the Alzheimer's study setup and what the concrete challenges that we currently encounter here.
Yeah, maybe I can, from my perspective, I'm a physicist and involved a lot in imaging part. So I'm a huge fan of imaging biomarkers, and I think that imaging biomarkers really can make a huge difference because they are able to pick up changes in the brain much earlier at a much earlier time before you see any clinical symptoms, and that makes it quite a strong tool for clinical trials. But what we do see in the existing imaging protocols is that there's a really huge variability. As Nicola said, there needs to be some organization, and there needs to be some standardization. And there, I think, is still a lack. This mainly comes, in my opinion, that we are hampered by what we can use and what is provided by the different manufacturers. You typically have to rely on their methods, and every manufacturer is providing slightly different methods to do an T1 weighted image or an T2 weighted image or diffusion weighted image. So when we are mainly looking at structural changes, that might be good, but as soon as we are coming to changes which are supposed to happen earlier, we have a huge challenge in combining those variable imaging protocols from different manufacturers.
Yeah, and
from your perspective, yes.
And to add to that, so unreliable data doesn't just create some kind of scientific uncertainty. It creates also a lot of regulatory risk. For example, when imaging protocols vary across sites, when data collection is not standardized, and when you cannot trace data provenance of every data point, regulatory agencies have legitimate concern and can put a question mark on trial results. Additionally, beside these regulatory uncertainties for physician and for the patient, unreliable data or scattered data or fuzzy data means uncertainty at the most critical moments. Just when the question arises, should we continue this treatment? What should we tell? Is the patient responding? We cannot really answer. So the main concern is that without standardized comparable data across time points and across sites, these questions become impossible to answer with certain degree of confidence. And this is definitely something that should be addressed in order to move forward in this disease area.
Mm-hmm. Absolutely. So when I'm listening to you, I kind of see two big topics, and that I also would like to now dive deeper into, right? On one hand, this importance of imaging biomarkers and also a development in imaging biomarkers, Matthias, to dive deeper in with you. And then Nicola, afterwards, I would like to talk with you a little bit more on this topic of variability that also seems such a central pattern here. But let's first still remain on the imaging biomarker. So, I mean, Matthias, clearly this is a core focus of yours. So why do you see these imaging biomarkers so as such essential, and where do you also see, let's say, the biggest opportunities going forward?
Mm-hmm. To your first question about the importance of imaging biomarkers, I think the real strength of imaging biomarkers is that they work regardless which pathway we are looking at. because we can look at changes forming in the brain via all the different pathways. And if we are looking at imaging biomarkers, we mainly talk about looking at structural changes. So the brain is really good in compensating loss of neurons and loss of gray matter to some extent. So you might not see clinical symptoms, but you will already see that there is something going on in the brain and that there is some loss of tissue. But the structural changes, even though they are earlier at some point, they can't not pick the very early phase, because at least in my assumption, I would assume first we have a change in function. There is no demand for certain brain regions, and then they will start to die. But in the beginning, there will be a functional change. There will changes in physiology. So, and now coming to your second part of the question, I think a huge strength in MR imaging, and that makes MR so special, is that we are flexible in the way how we create our contrast, and we do not have to rely on structure only. We can also look at function in the most general meaning. So we can look at blood flow changes. We can look at brain connectivity. So the whole physiology we can cover. And we see changes much earlier than structural changes. And that's a huge benefit, in my opinion.
Great. That sounds really, really like a fantastic way forward. So I'm really going to be curious also to see how Big Pharma is going to hopefully leverage this, and everybody who's in, let's say, for the race for a good treatment in Alzheimer's disease. Nicola, I already mentioned it, right? I think there was another big topic, and I want to switch focus a little bit now. The other big barrier, I think somehow both of you mentioned, but Nicola maybe a little bit stronger, is that of variability. So how can we address this challenge in Alzheimer trials?
So variability could be observed for multiple perspectives. Some kind of variability we cannot really control at the moment, so simply we don't understand it. But some variability in conduct and execution of clinical trial could certainly be addressed. For example, we already have some industry standards that are there for last 15, 20 years or more, like CDISC, that provide foundation for clinical data submission, clinical data interchange with regulatory agencies, DICOM standard that cover partially imaging domain. But what we really need, in addition, what don't exist right now, but add significant burden and significant variability in clinical trial conduct, is how we are operating, so how we are operational our clinical trials. So we need standardization on operational level. We need standardized workflow for how imaging biomarker trigger clinical decision, how adverse event data connect to imaging findings, just for example, how patient-reported outcomes are collected and verified. And then true standardization means building quality into the process from the really start. For example, real-time data validation, automated protocol compliance check, continuous monitoring of data integrity during data acquisition, data transfer, data processing. Because when you wait until the database is locked in clinical trial to discover those inconsistencies, it's already too late to fix them. The time have passed by. Patients are in the study that maybe shouldn't be in the study. Patients are not in the study that they should be in the study, and you cannot really go back in time and repair it. So better, let's concentrate and focus on standardizing operational aspect of clinical trials, because this is what we can tackle at the moment.
Yeah, I think you're really hitting a very important point here with once the images are acquired, you already fail in your first step, more or less, yeah, because also in the imaging part, you can only work with the images you have in the end. And that's why I promote since a while to also standardize on the imaging side, make sure when the data is acquired that the data you are getting is exactly carrying the information you want to extract later on, and that one is harmonized and standardized across the different vendors and across the different models, because it's quite surprising to see how variable the data is, and especially if you're looking into some very minor changes in the data, very subtle changes. You need to make sure that your acquisition part is just controlled and is fully under control.
Yeah. So, and I think, also this theme of standardization, that ultimately, right, is a very common goal between Mediri and Wemedoo. And I think we can openly say, right, that was one of the core reasons for us to enter a strategic partnership, because we kind of see the potential to leverage each other's strength on this topic. And maybe we can deep dive a little bit into that, because I think both of our organization have developed certain tools and which both can help, especially interconnected, to support more streamlined Alzheimer's studies. So, Matthias, maybe you could give us an overview of what you have built at Mediri. Yes.
At Mediri, we started very early on having a system which allows us to collect the data, do all the quality assurance, what Nicola already mentioned, and in addition to checking for the right protocol and stuff like this, also looking at the image quality. But for a while, we always relied on the data which was coming in. And if the data you're getting is of poor quality, you have this patient already in your study. So you can't really throw it away because you don't like the image quality. And that's why we started in collaboration with the Fraunhofer Institute for Digital Medicine, MEVIS, in Bremen. They developed a framework which allows to run MR sequences in a vendor-agnostic way. So you program the technique once, and then you have a driver concept which is controlling the hardware of the MR scanner to make sure that at every site, regardless which vendor you have, you're running exactly the same sequence. You're getting exactly the same contrast back, and you have more or less full control what's happening on the physics side. And as a physicist, I always like to control physics, or be under control at least. And then imaging biomarkers do not stop by acquisition and collection of data, of image data. You need to process the data, and especially modern imaging modalities which allow you to measure perfusion or other physiological parameters. They consist of a huge bunch of different images, and you need to process them to extract your final imaging biomarker. And this is also something mTrial can provide. So mTrial is just taking care of everything as soon as the images are there. And right now we have this interplay with this platform from Fraunhofer MEVIS, which allows us to really rely on very controlled protocols as well.
Good. And now, Nicola, how does Omnia fit into that?
So we have solved the problem from another side, and we built and designed the system specifically to solve the fragmentation problem. So instead of having electronic data capture, clinical trial management system, imaging management, ePro, eco-assessment on separate system. We simply eliminated need for integration and created a system where everything lives in one single unified, semantically defined database. When a patient completes cognitive assessment, data are immediately available alongside their imaging results, treatment history, and adverse events in one coherent view. So you are not waiting for data transfers, data reconciliation, data integration, corrections, and so on and so on. And collaboration between Omnia and mTrial represent exactly the kind of, let's say, integration and connectivity that industry needs. mTrial brings deep expertise in imaging workflow, imaging acquisition and processing, harmonization of the imaging protocols across vendors, quality control, and Omnia brings centralized package to understand and collect all other data under one hub and under one common shared meaning. So everything what we are doing is putting those imaging and imaging biomarkers into the clinical context.
Wonderful. And I mean, I could continue this conversation with both of you for quite some time, but unfortunately, we have the 30 minutes for this live stream. So I would have to start closing the session now to also allow for enough time for the Q&A. But as I do, I would like to just reflect and highlight a little bit what are the actionable lessons learned today and that we can all take away from here. So I heard about earlier intervention that trials can be more informative when we're focusing on preclinical or early stage patients and who are confirmed by biomarkers, and that is a challenge still to be overcome. But a biomarker integration, very important, using the imaging biomarkers, also physiological techniques to enable monitoring of disease progression and treatment response with much higher precision. That we standardize, we need to harmonize imaging protocols as well as workflows, and tools like mTrial, Omnia can help with that across the sites to ensure also reproducibility. We need to ensure efficient data management where these integrated platforms, Omnia, mTrial, can help on data collection, analysis, reducing operational burden, and so on. And last but not least, also adapting more adaptive trial designs, more flexible designs to lower the costs and improve responsiveness based on interim findings rather than hard designs from the beginning. So I think all of this together gave me a great understanding of where we currently stand on the Alzheimer's research. And I would be very curious also to repeat this conversation, let's say in a year's time, see what has changed since then. But as we wrap up and before we open a stage to questions from the audience, maybe one final thought from each of you. What's the one insight from today where you feel like that should really guide the next generation of Alzheimer trials? Matthias, do you want to kick us off? Somehow, Matthias, you are muted at the moment.
Sorry.
Yeah.
I might be a little bit biased on that, but I really deeply believe into imaging biomarkers adding a real additional benefit, and not only the structural, that's one part, but especially the physiological measures making huge difference. And I developed some of those methods during my PhD, which is a few decades back. But I think now that the time is really ripe to bring them into clinical trials and at a very early point see changes due to drugs on a very subtle level, but to make use of this subtle changes in the data, we need to standardize, we need to integrate all the data we have. And that's just important to be successful in trials.
Nicola, what's your one takeaway, one lesson?
From my side, very shortly, data quality and real-time data availability are not luxury, they are simply necessary. Next generation of Alzheimer trials and other trials, especially in neurology, will succeed or fail based on their ability to bring together high-quality imaging, biomarker, clinical data, and patient-reported outcome into one unified view. So we should stop treating those as separate data streams and start treating them as a part of a single patient story. And we will be finally able to deliver the insight that patient and clinician actually need at the point of care.
Wonderful. Thank you for this very clear statement. Good. And with that, back to the audience. I will look into my control panel if I have any questions. And I see we have one. The question is, what barriers do you see in implementing these solutions? My guess would be this is a little bit about, the new advances. Maybe we start on solutions and integrating these new biomarkers, Matthias, maybe in your direction before we talk more about the technology side of that question.
Yeah. So one really challenging part for the standardization directly on the MR scanners is that each manufacturer has its own way how to control it. And if you want to standardize it, you need to come to a point where you also integrate the different manufacturers. And we need to make them clear that it's in their interest to standardize different methods, because they are typically interested in somehow being different to the other, to the competition, and to provide some different tools. But we need to agree on some techniques across all the different vendors, and it is in their interest, but not every vendor actually believes in this, and that, in my opinion, is one of the greater barriers. And I hope that with the platform from Fraunhofer Mewis, we get some way to get into this in a quite simple way, and I'm quite optimistic. So we see quite some movement at different vendors right now.
Wonderful. And maybe from let's say the technology perspective, what are the barriers at the moment?
From technology perspective, the barriers are mostly in adaptation of standardized, meaningfully organized, let's say, operational aspects in clinical trials. So this is where we still have a huge gap, because simply we don't have standardized operational aspects around clinical trial conduct, organization, and analysis. We have from clinical part CDISC, but operational aspects are not covered, and we need really effort in these directions.
Absolutely. Good. I hope we sufficiently answered this question. Let us move to the next one. You mentioned the importance of treating early, needing the identification of patients in a pre-symptomatic stage. How do you imaging MRI, which is expensive and not always easily available, to overcome this dilemma? I would direct this to you, Matthias, as a question.
Yeah, so that's really a tricky part. So I think you don't want to set up an screening method based on MR. So we need to develop also maybe some blood-based biomarkers, which are maybe not they might be sensitive, but not very specific. So with MR, we could just focus on people who might have some signs in other biomarkers. That's in the long run. I can only concentrate on the imaging side and making the imaging as approachable as possible. And one way we didn't touch upon is one of the methods we developed is to use only the water in blood as an marker to assess the blood-brain barrier, for example. And so you don't need a contrast agent to apply, which also reduces costs, which also makes it easier to scan this. And there are techniques out, but you need to bring them to the market. And right now, the manufacturers are not interested in this. Yeah, but from my point of view, I fully agree with this question. I think imaging can only do some part, so you need to have some other tools which are cheaper, more approachable for first hint, and then or for first suspicion, and then you can actually have your MR imaging done.
And just shortly to add, on top of those, let's say, technical questions, there are a lot of discussion to be conducted about ethical aspects, about legal aspects, about different kind of those, let's say, directions that are going in direction screening, because this is separate area completely. However, it is really important to get an early diagnosis in order to really understand evolution of disease, of course, under all ethical scrutiny and all legal and regulatory aspects. But this requires separate, let's say, webinar dedicated only to this topic.
Which might be a good follow-up for us to consider here. Thank you for the question. Let's move on to the next one by Christopher Ilgen. But aren't the scanners qualified before being used in the trials?
Yes.
Matthias, I will let you answer this, but this is frequently so misunderstood question. And I'm also from radiation therapy part. I'm radiation oncologist by myself. I have spent 70 years in this domain. We really have to try to explain to people what DICOM is and where are the challenges and what actually device mean and how this is processed and software and everything in very details. But this once more requires separate webinar. But please, Matthias.
Yeah, maybe right quickly. So the DICOM is the format for medical images. So all medical images are stored in a standardized format, which is called DICOM, which includes some information about the protocol which was used to acquire the data. And when I started Mediri, our first product was quality assurance. And I thought, okay, the main feature should be checking for image quality. That was my specialty. But then I included also checking for the different DICOM headers. And I was surprised. So even though the scanners were qualified and were checked quite hardly once, the protocol seemed to be changing in some at some sites, and I don't know why, but it picked up quite a lot of changes. And I think the main reason might be that people are not aware of if you include all different scanners, you always have to actually design your protocol to the weakest scanner or the scanner with the least performance. And so the others could do better. They could create more images, better images, faster images, but it's not standardized anymore. And then the next part is even if you have all those DICOM headers in place, it doesn't mean that the same physics is actually played out on the scanners. So each manufacturer is doing different stuff, which is not documented in the DICOM headers, unfortunately.
And are proprietary and cannot be tackled, cannot be easily changed. There are some rightful reasons for that, but there are some also other reasons. Anyway, we need a little bit more effort in understanding and in achieving those standardization aspects.
Absolutely. But thank you very much for this great question. Certainly hits a nerve. And then last but not least, we have one more question before we close the session, cognizant that we're already 5 minutes above time. But how do you see AI influencing imaging workflows in Alzheimer's studies? And what impact would we be able to see?
Depending. because AI is really powerful tool or method, consists of various, let's say, mathematical models. But this is then once more chicken egg problem. If you then don't have reliable data at the beginning, you cannot really create reliable AI based on it. And I don't think that anytime soon we will see significant impact of AI in imaging in Alzheimer's disease, simply because of all those reasons that we have expressed earlier. So it is really powerful. This is the way to go, definitely. But the road to this promised land is paved with a lot of obstacles.
Yeah, maybe I can also add. I fully agree with what you said, and would add: if you have rubbish in, is rubbish out. That's definitely true. But to be honest, AI is really good in fooling you. So even if rubbish is getting in, you are getting something out which looks like a really precious information, but in fact it is rubbish, but you don't realize it. And
you
that's
cannot
really the challenge to make sure that that you can trust AI. Yeah, and that's some effort we are also working on, of course, because I think it is a powerful tool, but you have to be careful what's getting in and what you are get out and how to interpret it.
Absolutely. And I think that's a very important insight that we can probably take away even beyond, let's say, the scope of this conversation. But yeah, thank you all for your questions. Thank you both for the insights. I certainly take a lot away from today. And yeah, I'm hoping to see both of you for a follow-up. That's clearly out now in the making. I think there were many topics we identified, and also to everybody who tuned in to see you in the next webinar next month. So thank you all, and have a wonderful rest of your day.
Thank you very much.
Yeah, thank you.